Thank you, Frank, for the nice introduction. Thank you, everyone, for being here and listening to our story. At cellvie, we're working on mitochondrial transplantation, so to leverage the therapeutic potential of mitochondria. And I appreciate that for most of you, mitochondria don't need an introduction. After all, they are one of the hallmarks of aging, or rather, their demise is. But I still want to make sure that everybody understands the pivotal role that mitochondria took in the emergence of complex life. Billions of years ago, a bacteria at the time, they did merge with what was, at the time, a simple cell organism, barely surviving, we would probably not call it alive today. But it was that merging of the bacteria and that organism that brought about a symbiotic relationship. The organism would provide the bacteria with an environment in which they could thrive, whereas the bacterium would give them energy. Energy that would allow them to evolve and develop the functions that we too enjoy, every one of us today. So mitochondria are found within almost every cell of the human body. And in the course of enabling our evolution, they've taken on a number of pivotal roles. The most known, of course, is their energy production, ATP. But that's not where it stops. They're responsible for various signaling pathways. They're involved in the apoptosis and cell differentiation pathway. Whenever mitochondria get damaged, cells die or lose function. There's more than 50 different diseases that are associated with some form of mitochondrial malfunctioning. But we've found a way to use these little organelles to our advantage. But before I elaborate, I'd like to share the story of Avery. Avery was born with a complex heart defect. In the procedure to save her life, she sustained a prolonged lack of blood flow, kind of like a heart attack. She went into a cardiogenic shock and had to be connected to ECMO, these heart-lung machines that got a lot of attention during the COVID times. These machines artificially oxygenate and pump the blood where Avery's heart was not able to. Multiple days on ECMO, there were very slim chances of her survival. Fortunately, Avery was enrolled in a clinical trial ongoing at Boston Children's Hospital. She received mitochondrial transplantation to ameliorate the damage that came from that lack of blood flow. Today, she's a happy and healthy 5-year-old. Or, to quote her mother, she's a rock star. Avery's condition is called ischemia-reperfusion injury. It sounds complex, it's simple. It arises whenever the flow of blood is interrupted and subsequently reintroduced. You will all know the medical conditions associated with it. Heart attacks, stroke, organ transplantation, long surgical procedures such as Avery's. Whenever the flow of blood is stopped and reintroduced, damage ensues. What few people know, though, is that this damage that arises during ischemia, which is that lack of blood flow, is a damage to the mitochondria, to the core energy-producing factory of the cells. They turn the oxygen that's transported via the blood into energy, allowing cells to maintain their function. So when those mitochondria are damaged, as we learned earlier, cells die. So they do in a heart attack, during organ transplantation, in any other moment where your blood flow is interrupted. But what if you could replace and augment the mitochondria that are damaged in conjunction with a heart attack? What if we could reinvigorate that interrupted cell energy metabolism? That's exactly what Jim and his colleagues have shown to be possible. cellvie's approach rests on therapeutic mitochondrial transplantation. The product that we are developing is off-the-shelf allogeneic mitochondria that are derived from a cellular source. We found a way to treat them and to ultimately freeze them so that they can be seamlessly integrated into the clinical workflow. We have found various ways of delivery, including direct injection into the site of injury, vascular delivery, but also aerosolized delivery of the mitochondria. Once they are injected, we've shown that they are integrated by the cells. It seems that they retained that bacterial origin in the sense of their ability to transfer through cellular membranes. Endocytosis is the process by which they're integrated. You can see here on the right-hand side where they go. The green are mitochondria expressing a GFP signal. The red is the mitochondrial network inside of the target cell. The blue is the nucleus. And as you can see, the mitochondria don't go just anywhere. The mitochondria seem to merge with the mitochondrial network inside of the cell. And that's how we believe they're able to contribute actively to that cell energy metabolism. Specifically, we believe they rescue the cell energy metabolism. So you can think of it like jump-starting a car. Through the heart attack, that energy metabolism is interrupted. Through the mitochondrial transplantation, it's reactivated, allowing cells to perform in their normal function again. The technology was developed at Harvard Medical School, and as Frank rightly said, Jim McCauley is here today. So if you're really excited about this subject, you should remember that face and find him, because he's the one who came up with this concept. And he came up with it because he was frustrated that none of the agents that he was using would work on mitochondria. Mitochondria are extremely difficult to treat. Their disease association has been known for decades, yet few, if any, therapies to treat mitochondria exist today. Our other two co-founders are Drs. Del Nido and Demani, who were the first to bring this technology into the clinic together with Jim, initiating that trial that saved Avery's life. I'm a repeat entrepreneur in the medical space, having started, built, and successfully exited a company called Adhesives Medical, developing surgical glues. And together we set off to bring mitochondria to the patient. I want to share what impact this therapy can actually have. That's data from a pig model that Jim conducted, mimicking a heart attack. What he has shown is that with mitochondrial transplantation, the damage that ensues, so the dead muscle, can be reduced by 60%. How that may look like, I'm happy to illustrate with a bit less of an invasive model. This is a mouse that was subjected to 2 hours of hind limb ischemia, so no blood flow to that hind leg. As you can see, basically the leg has lost its function. With mitochondrial transplantation, on the other hand, that function is largely retained. It may be that we've found the first ever true treatment for ischemia-reperfusion injury. Given the solid safety profile, as well as the grand efficacy, is what caught our co-founders' attention in their hope to find an agent to help patients such as Avery, patients on ECMO without treatment options. And so they initiated that clinical trial at Boston Children's Hospital that is still ongoing. And they conducted a comparison against a historical control group with the first patients, showing that 80% of those patients achieved myocardial recovery, which we basically define as their ability to separate from ECMO. In that historical comparator, it was 29%. So there's some real-world data on the potential impact that mitochondrial transplantation may have. At cellvie, we're seeking to bring about mitochondria as a new category of medicines. We seek to build a mitochondrial powerhouse that enables mitochondria to reach patients in various indications. And we truly believe that there's 3 pillars to our potential success. The first is that we need to optimize mitochondrial preparation, high yield, a safe, reproducible product. We also are pioneering the development of off-the-shelf mitochondria, as I explained earlier, so that they can seamlessly integrate into the clinical workflow. And finally, we're working on a pipeline of applications. We truly believe in the potential of mitochondria as a platform and seek to pursue it as such. And we're very fortunate to have Kisu as our investor, allowing us to pursue these mitochondria as a platform, specifically seeking to bring them into the field of aging-related degeneration. And I alluded to it in the beginning. It shouldn't come as a surprise. Their demise is associated as one of the 9 hallmarks of aging. And we believe that mitochondrial transplantation may help in mitigating and maybe even reversing some of the mitochondrial damage that ensues across the aging space. we hope to be able to achieve higher ATP availability in those cells, to lower ROS, to restore CO2 hemostasis. We really believe mitochondrial transplantation can help in what is a very complex cellular, so to speak, mechanism. And in order to better understand how the mitochondrial transplantation could help, we looked at 4 different aging-related degenerations and tried to better understand how does the mitochondria, let's say, demise affect the disease pathology. And how does this compare to what we've seen mitochondrial transplantation to be possible to affect? We came to the conclusion that there is a number of, let's say, very solid evidence linking mitochondria to the disease pathologies of neurodegeneration, muscle degeneration, hearing loss, skin aging. Each of them clearly have a mitochondrial link. But not all of these links were equally strong or equally well suited towards the effects that we had seen mitochondrial transplantation to bring about. And so we decided to focus on muscle degeneration as our first indication in the aging-related degeneration space. And we've made some progress. We've started with just a proof of principle study to show, does in aged mice mitochondria affect their ability to perform? And I wish that next slide would be showing you the data thereof. It won't. It's data I cannot yet share, but let's say we're very encouraged by what we're seeing in our pursuit of muscle-related degeneration. cellvie is about to enter its next big phase of development. The seed phase is coming to an end with what I believe very meaningful milestones that we've achieved as a company. We've been able to turn what was a therapy that was autologous, freshly isolated mitochondria into mitochondria that come from cells and may be able to be stored for months, if not years, to seamlessly integrate in the clinical workflow. We are about to start our large animal trials to show that these off-the-shelf mitochondria work as well as the fresh autologous ones. We've shown that our pipeline exists and that we may treat one of the most pervasively present diseases, which is aging. And so we're seeking to take mitochondrial transplantation to the next level. We're seeking to raise 35 million in our next funding round, which we will kick off in Q1 next year. Part of that funding will go to manufacturing, enabling a production of mitochondria at scale. We will push our first program in heart disease into the clinic and then expand our pipeline specifically and primarily in the aging-related disease space. I can say that I'm happy to report that we are not alone. Mitochondria as a space has been gaining a lot of traction over the last 5 to 10 years. Big deals are being made, and we're happy to be a part of this very vibrant and emerging ecosystem of using mitochondria as a drug, as a therapeutic modality. There's one critical question that motivates each and every one of those companies on that slide, including ourselves. And that's the question of, what if mitochondria had more to contribute to the evolution of mankind than igniting it? Thank you.
Thank you. Fascinating talk. I think we have time for a question or so. So I give it to the audience. Somebody wants to ask a question. There are two. So. Stephen, yeah. Okay.
I was afraid Stephen was asking a question.
So yes, with that last slide, you left us hanging a little bit. So there's a lot of competition in this space. How are you differentiated?
I mean, there's a number of aspects that I believe work in our favor. On the one hand, I think that the space of mitochondria-related diseases is huge, right? So it's never going to be a winner-takes-it-all market. So that's one aspect which I look towards very favorably. And the second aspect is that the companies don't all do the same thing. Right? So if you take Menovia Therapeutics, for example, they're using mitochondrial transplantation to amplify the function of stem cells to then treat a patient systemically. Whereas we use directly naked mitochondria in an effort to treat cells within the body that have been affected due to ischemia or due to age ultimately. So I think that each of us will carve out eventually its own space, but it's important that we all work towards it so we get it away from the fringes to make sure that investors step up to fund the evolution of the technology and large pharma takes note.
Okay, one more.
Thanks for the great talk. I found it fascinating that it's actually possible to replace mitochondria in an adult, or children in that case. So if mitochondria are really responsible for a big part of aging, then of course a very promising approach would be to totally replenish the aged mitochondria in the body. Have you looked whether the new mitochondria which you introduce actually outcompete the older or innate mitochondria?
It's a very interesting question that has not an easy answer. But one answer I can clearly give is that the possibility of just replacing the entirety of the mitochondria we consider do not at the moment as feasible. And the way that we currently have seen the mitochondrial transplantation to act in the ischemic perfusion injury setting, for example, is that the new mitochondria basically help the present mitochondria to perform in accordance to, so to speak, the pre-ischemic phase, so bringing them back to normal function. So it's more about, let's say, re-establishing the mitochondrial function within the cell rather than replacing the mitochondria in their entirety.
Thanks.
Thank you. Okay, if there are more questions, please confer them to the network room.
Yeah.
And give a big thanks to Alexander.
Thank you.